this podcast, you'll find interviews with high-performing, successful individuals in life sciences. On a weekly basis, we cover their proven methods, principles, strategies, and mindsets to implement new technologies that scale to meet the needs of people in our world.
This episode of Life Science Success is brought to you by 5280 Life Sciences Consulting. If you need help setting your strategy, meeting your operational goals, or you are selecting and implementing critical technology in your business, we should talk. Visit 5280lifescience.com to schedule a meeting and find out more.
Today on the podcast, I'd like to welcome Ray Price. Welcome, Ray.
Hey, thanks for having me, Don.
Can you tell the audience a little bit about yourself?
Sure. Born in Knoxville, Tennessee, and grew up there. Went to college in southeastern Washington, a little tiny school called Whitman. Did biochemistry major and then went to grad school at Vanderbilt for my PhD in pharmacology. When I finished there, I moved to Japan and worked as a research scientist for a company at the time was called Fujisawa. Now they're called Astellas. They merged 2005. So worked as a research scientist there for 4 years and really enjoyed the ability to interact with key opinion leaders. We actually had a drug that moved into phase 2. So pretty fortunate as a researcher to be able to do that. And we even participated in designing clinical trials and things like that. Really great experience, great coworkers. 2005, though, after the merger, we had to move within Japan and start over with the network and community. And it was a little more difficult than we anticipated. So I ended up coming back to the States and joined a company called Acusella, where I was for 2 years. And we had the CEO was Japanese, and all of our funders and our partners ended up being Japanese pharma companies. So that's where I moved into business development. And we partnered a platform technology to do retinal drug discovery and retinal drugs. And then moved to Paris, France for 2 years, joined a startup there. I worked on something actually I'd worked on in grad school, so knew the science part of it, but that was really more partnering and business development of a diagnostic. I did my MBA the second year there at a school called INSEAD, one of the quite good school, I would say, not done in French, but we had campuses in Fontainebleau outside of Paris, Singapore, and Abu Dhabi. So got to spend time on all three campuses. And then, came back to the States and have been in San Diego a little over 10 years now, working for a series of CROs, company like we'll talk about, I think, a little bit, company called BioSeek that got acquired by DiscoverX, that got acquired by Eurofins. And then I moved back a little over 2 years ago to a company called Phenovista that we do high content screening and fluorescence microscopy as a service. But all of that has been in business development since the Seattle company, basically.
So how did you make the jump so you went from school to Japan? That's an interesting leap. So how did you make that transition?
Thankfully, Al Gore had invented the internet by then. And so I went through and found companies in Japan that had, I basically just went through papers and found senior authors at Japanese pharma companies and said, I'm looking to graduate and my degree will be pharmacology. And I should say I had a summer experience in Japan. So is sponsored by the NIH and the Japanese version of that called Monbusho. And they had about 60 grad students that went to Japan for 3 months. And so I worked at a pharma company too called Eisai. So I really got a chance to figure out at least a little bit what it would be like to live there and fell in love with the country and the people and said, I want to go back for that as part of my career. And so, wrote to probably few hundred different PIs and things of these companies and had 4 interviews, 2 in person. One, they flew me to Japan actually, and one was in Chicago, and that was where I ended up going. But it was like sight unseen. I'd never met any of my coworkers or seen the lab or anything like that.
Wow. Amazing. It's great to get to go from, so you're studying somewhere and then you find this path. So in your global experience, did you learn any other languages?
Yeah, my Japanese is the better of the two. I can listen to French okay, and my time there was around the 2008 recession, and so especially the second year before I started the MBA program was a little uncertain. So my French is not as good as my Japanese, but for American, our expectations are quite low at being multilingual. So I can absolutely exceed those. I know a bunch of jokes in Japanese.
The one thing that I really remember from traveling to Japan that kind of surprised me was that 75% of the country only speaks Japanese. And
Yeah.
that was surprising for me because I just envisioned that in Japan, especially, that whenever you travel there, that, hey, look, there's no real danger of running into somebody that only speaks Japanese. But lo and behold, they warn me, actually, being a global traveler, is make sure that when you check into the hotel, you get their business card, because if you get lost,
totally.
one thing that you can do is hand the taxi cab driver the business card and say, I want to go there. So.
Yeah. Well, and the address, I mean, yeah, that's all those things. The addresses aren't linear like here. It's not 1357. It's the order that the buildings were built on a block. So it can be completely, disorienting. And that was actually part of I took classes when I was there for the summer. And then when I first moved there, the company actually paid for me the first year to take lessons also. And yeah, they had a kind of triage plan, though. They said, if you're desperate and need help, find a college-age woman, a man. It was like a rank order of likelihood that they would be able to communicate in spoken English, because written actually was probably a little bit better, because that's what they studied. But contractions and colloquialisms and things were really tricky.
Absolutely. So you spent, looking at your sort of online profile and things like that, you spent quite a bit of time in business development, BioSync, and then Phenovista. It seems like you spent a lot of time helping businesses connect with potential opportunities. Can you tell us a little bit about what led you to that area from your education?
Sure. Well, so I'd done the PhD in pharmacology and worked in drug discovery. So I knew, at least at some level, what the pressures and the conditions for working in that environment were like. And I had experience, like I said, moving a compound from preclinical work. We submitted an IND and moved that into a clinical study. I had a kind of linear experience, at least with a part of the process. And then at AccuCell, at this company in Seattle, it was a bit of the right place, right time. And they had a VP of business development who was a lawyer, so not super technical, but really good at negotiations and presentations. But he wanted someone that could partner with him on the technical side. And so I'd worked in that platform for a few months already, and so knew it, reasonably well, let's say. And so it was that the buddy system, let's say, for being able to go out and present. And that's where I really, that kind of mentorship role and relationship was really key to me succeeding in that. And subsequently, each place I've gone, I've learned something about the process of, negotiation or contracts. And doing the MBA also helped for that piece. Course of how do you interact with people, how is power distributed in an organization, all of those things that they don't teach you in PhD school.
Absolutely. Yeah. It's funny because a lot of people that I've talked to in terms of like career coaching and things like that, we've had these discussions about, hey, I do this today and I want to do something new tomorrow. And typically the way the conversation at least goes, if you have it with me, is the chances of you just randomly moving into some other area that you've never done anything in is zero. I think the leader would truly have to take a gamble on you. And it's almost like flipping a coin. Do I think that the person's going to work hard enough? Are they going to really try and get into this thing and figure it out? Or are they going to fail? And then I'm going to wind up having to do all their work. And at the end of the day, probably have to make an adjustment in some way. I try and tell people finding those mentors or finding somebody to help you bridge is really critical. So it's great that you found that, especially to bring you to something that you've tied to throughout your career. So that's great.
Yeah. Yeah. It turned into that anyway. Like you said, at that time, I wouldn't have, or even in Japan, or in grad school, I wouldn't have said, in 5 years, I'm going to be doing business development for technology platforms or clinical assets. Being open to that sort of concept, and they at the company recognized that as well, of my interest. It is a very low bar among scientists to be extroverted, but being able to interact with people and knowing the language in Japan and the culture and being able to, that was a tremendous part of why they hired me in the first place, but then also were open to moving me in that kind of an untested role like that.
Excellent. Phenovista is described as an imaging CRO. Can you describe a little bit about what they do and who your primary customers are?
Sure. So it's mostly pharma and biotech companies. And, typically it's some version of this scenario where we take normal cells, let's say normal neurons, and we have a couple of phase 2 SBIR grant to look at this. So nothing confidential here, but take a normal neuron, cortical neuron, a disease neuron from an Alzheimer's patient, iPSC derived, and often they look different under the microscope. And then we test for compounds or antibodies or gene therapies or cell therapies that can make these Alzheimer's guys look normal again. Okay. And we can do that and use a microscopy. So, we're staining and imaging and analyzing and quantifying these differences, and then being able to help companies make decisions about their programs or compounds to move forward or not. And
so with that output, that's okay. So with that sort of output, so then the drug company or the company that's more or less working on the treatment, then they use your sort of output for trying to figure out or sort through what the best pathway forward is, if I hear you correctly?
Yeah. Yes, that's one potential outcome, or whether that's which of these 50 drugs is the best one in terms of the most efficacious or most potent. We've helped companies triage gene therapies, right? Which of these vectors is the best to approach or has the most impressive neuroprotective effect. We work in immuno-oncology. So it really is pretty broad. One of our distinctives, I would say, is the flexibility that we have in being able to run a range of different assays and take a kind of core capability and apply that to a new disease area that we might not have worked in.
Okay. With confocal imaging, can you describe what types of things that can be seen in these images?
Yeah, so most of the time we're looking at cells and either subcellular structures, right? So there's a technique called cell painting that we use a lot. That's essentially a set of dyes that will label different cell compartments, nuclei, cell membrane, ER, ribosomes, and be able to, it's a similar concept to what I described with Alzheimer's, where you can take a normal cell or an untreated cell and then a cell that's treated with a particular drug, whether that's a toxicant or a drug that's improving cell health. And you can match those two, or you can take the sort of drug-treated cell, generate a series of measurement outputs from those dyes and from those images, and be able to say, yeah, this cell looks healthier than the other. And we can even go as deep as matching using machine learning and other techniques, the signatures that are present in those treated cells with other drugs. So someone could send us a drug that we don't know anything about and say, oh, it does, the pattern matches, or the fingerprint or the signature matches this other drug. So it probably is working the same way.
Very interesting. And the one thing I really remember, having spent a little bit of time in this space, is just the variety of all of I don't have the same trained eye that you or somebody else that has looked at a lot of these images has, but just the sort of artistic view of some of those images. They just can be amazing in some cases, all the structures that can be seen and the colors that do come out and things like that. It's just an amazing sort of thing to see as well.
Yeah, no, for sure. It's part of what's fun about what we do is they always say pictures tell us that, or take the place of 1,000 words. And for us too, it certainly is exceptional, some of the images that people can generate and collect. And for us, it's actually also turning that into a quantitation and being able to have it be something that someone can use to make a decision on. And that part's not so trivial either. But yeah, a lot of companies we worked with have taken what we've given them, and for investors, for example, where if you can show, well, this is a normal healthy cell and this is a really sick cell, and this is a cell after our treatment, it's pretty compelling. And if you have a bar graph, that's great too. But the ability to really visually say, oh yeah, that's a pretty clear difference in what you're doing.
Yeah, it's one of the things that I've talked a lot about in quite a few podcast episodes, is that the one thing that I think that we could all do better is just in that ability to tell a story better. And a picture definitely does help tell a story. You can point to something and clearly say, you see the difference between this one and this one. And maybe even an untrained eye could maybe even see the differences between something that looks more healthy versus something that's not.
Yeah.
So
usually it's pretty clear, or we like it the best anyway.
Yeah. So given the new technologies that are out there between next generation sequencing and molecular PCR and things like that, can you describe from your perspective where you feel like imaging fits in the landscape of life sciences instruments? Sure.
We're part of a broader strategy, let's say, for drug testing called phenotypic profiling. So instead of what might be a genomic, if a target, for example, is important in cancer, right, KRAS or something like that, you can go in and design drugs to target that, and then your assays, or tend to be focused around that particular target. What we do is more broadly known as phenotypic profiling, and so it's basically a phenotype or an appearance of something, right, of the cell, whether that's cell health or something like that. So historically, anyway, in the drug discovery industry, more drugs have actually been discovered with that approach than with the target-based approach, at least initially. After that, once people know that this particular target produces a phenotype, then they'll move into a target-based approach or strategy and be able to say, yeah, we can make a more selective molecule for this, or more potent, and it will be even better, the second time around than the first time. And within a lot of molecular biology, for example, is geared towards that, like of looking at knockouts, CRISPR and things like that, of looking at knockouts of a particular target, or how does my drug treatment change this particular target. Whereas what we do is, first of all, it's proteins typically. So it's really the kind of downstream consequence of those gene or genetic changes or messenger RNA changes. And phenotypically, we can also often pull up things that may not be known, or we don't need an a priori hypothesis of what to look at specifically in those cells, where you often need that in a molecular biology technique.
So in today's environment with COVID and things like that, I would imagine you have a lot of people that are having to spend a fair amount of time in labs, and you probably have more samples that are sensitive to, trying to get them imaged before they change or anything else. What have been some of the greatest challenges you've faced recently?
Yeah, certainly, especially this in the last couple of months, like supply chain things that we didn't have to deal with before. I mean, even just ordering plates and sort of basic reagents. Certainly, if we were doing qPCR, we'd be out of luck, and all of those things are going towards COVID tests and things like that. So that's one. Sourcing, a lot of work that we do is with primary cells, which means taken directly from people or patients, and that has slowed down tremendously as well, because people aren't getting elective surgeries or not going in to see their doctor or being treated, clinical trial enrollments, things like that. And then certainly the safety and welfare of the lab and operations people at the company too, and we're in California, so trying to manage what the state tells us we can do in terms of the number of people. And I've been in maybe 3 or 4 times since March. There's definitely some trade-offs of being virtual versus being in the lab or the office, and protocols to keep people safe there, because of course that's the main priority.
So have you seen any main benefits overall in terms of having to work virtual as well?
I would say for us as a company, because we do services, it's actually been a good season, because people have restricted access to labs. So they're using the time that they have to do really core priority projects. And so we're maybe the trickle down of that, and because we'll work with outside cells and materials, people have been sending us projects that we might not have gotten otherwise. I'm frankly a little bit tired of talking to my computer. I really like the low bar for the scientists being extroverted, but I do enjoy traveling and meeting people in person, and I'd be very pleased to have those things come back. But there's a lot of people that have it, worse, and we're very grateful that we still have been able to operate and even grow. We were 12 people at the beginning of the year, last year, and we were 25 in December. So, from that point of view, it's gone good. And all things considered, I'm very grateful to be, doing what I'm doing, and that the company's still been able to continue to grow as it has.
Yeah, it's interesting how we found lots of entrepreneurial ways, or lots of innovative ways to solve some of the key challenges that obviously require some level of human touch, especially in these lab-based things. And then you have to your point, the human touch portion of all the other job functions was removed. And I think numerous guests here have said, hey, look, I definitely look forward to getting back to shows, or getting to be able to have coffee or drinks or whatever with people as well. Yeah.
For sure. And I mean, I think this was, I think early on, like in the summer and late spring last year, I remember reading about for kind of continuing business or expanding from an existing customer. I think that's probably maybe not as dramatically changed, but building new business and people that you haven't met. And they were talking about it for entrepreneurs and fundraisers, and like JP Morgan, we're doing that next week, but it's all virtual. or Society for Neuroscience is also next week in virtual. These are meetings or conferences that we would normally go to and find both meet with existing customers and build those relationships, but also new ones. And yeah, I think knock on wood, the vaccine rollout will continue to go well, and maybe in the fall things will start to return, maybe back to normal. But I would say it would be tremendously difficult if we were starting up during this season, and or that there wasn't a kind of existing customer base. I've been doing this, like you said, for about 10 years, particularly in phenotypic assays. There is a customer base and existing relationships that I can pull from, but it would be tremendously difficult to try and start from scratch from a brand new greenfield project like that.
And thankfully, the technology was somewhat in place. If you go back to your earlier comment, so if we were in times before Al Gore created the internet, then it'd have been a little bit more difficult, because all of a sudden we would have been trying to figure out how do we communicate with each other without being. So you would be on the phone with no face. So,
for sure.
Yeah. So there are 3 questions that I like to ask every guest. What inspires you?
I love, you went back this idea at the beginning of, or in the intro, when we were talking pre-show anyway, the idea of being able to match people with needs up with people that can meet them. And I think that's why I love doing what I do in business development. In grad school, it was helping people move, or even now it's helping people find jobs. That's just, and if it can be a technical problem that a client has that either we can solve, or I can say it's not something we can do, here's a referral that you can use to meet that need, or that's the fun part of what I get to do. Excellent.
And what concerns you?
So especially in the pandemic, I've been trying to, I think about this idea of having a sphere of influence, things that I can change and do something about or do something to, and the sphere of concern. And that the closer that those two things are, and this is not my own idea and I cannot come up with the reference for it, but this idea of being able to match, the closer that those two things are, your sphere of influence and your sphere of concern being overlapping. The things that I have 2 kids that are in college. My daughter's in her senior year of high school, and then I have a sophomore in college. And those things of her senior year has been a little bit lame, frankly. And what can we do about that? But at the same time, we're safe and healthy, and she's picked college, and there's a lot of people that have a little asterisks by 2020, 2021.
Yeah, it's got to be an interesting time. My kids are all a little older and out of the house at this point in time, but similar sort of thing. You'd be concerned regardless that one, are they getting the experiences that they need to have? And is this going to at all hinder any portion of their development or final career trajectory? Yeah, definitely a good point. What excites you?
I guess it's also back to that earlier question. I love doing what I get to do and matching people up and talking about doing this kind of thing like career advice and, input. And yeah, those things are what's fun. Sending the elevator back down. Again, not my statement, but send the elevator back down for the next generation of people. Those are things that I love doing.
Really good. So Ray Price, thank you so much for being on the podcast. I greatly appreciate you being here.
Yeah, thanks for having me, Don. Appreciate it very much.
Thank you for listening to Life Science Success. For complete details about this podcast, including show notes, how to get in touch with guests, and more episodes, please visit www.lifesciencesuccess.com. If there's someone you'd like for us to invite to the show as a guest, please let me know by sending me a message at the podcast website. Please click subscribe on your favorite podcast app, share the podcast, or tell a friend about it. And last but not least, rate the podcast. Thank you again.